and be with us today for spending for the benefit of our students and for the faculty member. Thank you, Mr. Debanjan Das. Now I request Mrs. Lara, Madam, to give a brief introduction about our college and the Department of Pharmaceutics.
Okay, sir. Good evening and warm welcome to all delegates and participants for the International Webinar Series. First of all, I would like to thank our beloved principal, Dr. A. Meena ma'am, for giving this opportunity. It's my pleasure to introduce briefly about our institution. KK College of Pharmacy was established in the year 1992 under the able leadership of Professor K.R. Arumugam sir, who is an educationalist, academician, and served as a member of Senate and County Council of the Tamil Nadu Dr. M.G.R. Medical University. He is a constant source of inspiration for us. Our Vice Chairman, Dr. A. Babu Datamani, who always ensures the required resources of our college. Our Principal, Dr. A. Meena ma'am, is very humble and playing a key role in success of our college. Our Vice Principal, Dr. A. Shanti ma'am, who is very dedicated and provides valuable input for continuous improvement. Dr. Vaidalingam, Director of our college, he provides great support and backbone for the progress of the institution. Dr. Sendil Kumaran, Dean of our college, who always encourage and motivate all staff and students. The institute grew from strength to strength, starting with diploma, degree in pharmacy, and gaining the full-fledged pharmacy college status, including Master of Pharmacy and PharmD program. KK College of Pharmacy is affiliated to the Tamil Nadu Dr. M.G.R. Medical University and approved by the AICT and the Pharmacy Council of India. The college is located in a green environment at Gargambakam Poru, Kundathur Road, Chennai. The Pharmaceutics department has well-spacious separate laboratory for UG and PG students, which is also well-equipped with latest instruments like rotary tablet punching machine, ampule filling machine, and stability chamber. Our faculty member has published various research papers in national and international journals. Our college has started conducting a series of webinars. We have successfully completed 12 webinar series by various departments of KK College of Pharmacy. Today, the Department of Pharmaceutics is hosting the second international webinar series on the topic Quality by Design, Industrial Approach. Thank you, sir.
Thank you. Thank you, Mrs. Lara. Now I request our energetic Vice Principal, Madam, to give a welcome address to the audience.
Thank you, sir. Warm welcome, everyone. It's my great pleasure to welcome you all. Thanks for joining us for today's webinar. Well, we are lucky enough to be able to reach out the speaker and you all, even during this pandemic. By the way, we have been conducting the webinar series. And I'm proud to say that this is the first international webinar organized by the Department of Pharmaceutics. Well, today we have an eminent speaker, Dr. Debanjan Das, from United States of India. To be frank, I was awestruck looking at his profile. His vast knowledge, vast experience in the field of pharmaceutics. So I'm 100% sure that we are going to have an informative session today. And definitely that will pave the way to those who are looking for an opportunity to work in the industrial field. At this juncture, I would like to welcome Dr. Debanjan Das. It's my great pleasure to welcome you, sir.
Thank you so much.
Yeah.
Thank you so much for having me. I apologize, I was a little bit late because I was just sorting out some things because it's morning time for us. But let's get started. Hopefully, I will be able to give you some good seminar.
Yeah.
Okay. So I will share my screen from my side. Is it?
1 minute, sir. 1 minute.
Okay.
We would like to have an introduction about you, sir.
Okay.
Before that, I once again would like to welcome each and everyone on behalf of KK College of Pharmacy and its management, our beloved principal, Meena ma'am, and faculties, and our students. Thank you, everyone. Over to Sendil, sir.
Thank you. Thank you, madam. Thank you very much. Now I request our Associate Professor, Dr. K. Karthik, to introduce the speaker to the audience.
Good evening, everyone. I deem it a great pleasure and honor in introducing today's speaker, Mr. Debanjan Das, who is multifaceted and is an expert in modern QS and manufacturing concepts. Mr. Debanjan Das has completed B.Pharm from Rajiv Gandhi University of Health Sciences, Bengaluru. He was awarded Master of Science in Drug Delivery from St. John's University, New York. He also possesses Master of Science in Industrial and Physical Pharmacy from Purdue University, Indiana, US. He holds a certification in FPGEC from National Association of Boards of Pharmacy, Illinois, USA. He also holds other professional certificates from various universities. Mr. Debanjan Das started his career as a project manager in Progressive Group Kolkata, where he developed integration of manufacturing steps which led to cost saving. While pursuing his MS in drug delivery, he worked on targeted CNS drug delivery of biologics, and also he developed the world's first human blood-brain barrier cell line for rapid drug screening. As a research fellow in Purdue University, he successfully developed solid dosage forms involving specialized techniques which minimize manufacturing steps and reduce overhead costs. He was a senior formulation scientist in Smithers Pharma, where he worked on nanotechnology to deliver proteins and peptides. He was also responsible for a number of NDA and ANDA submissions during that time. He steadily climbed the ladder of success as a lead product and process scientist in contract pharmaceuticals, where he employed QbD principles to get technology transfers done efficiently. His expertise in using the tools of QbD helped to fast-track product development by strategic planning and overcome formulation challenges during his tenure as application development manager in Dow Chemicals. As a manager in formulation and process development in Fleet Labs, he was actively involved in new products development and improvements and product line extension of existing brands and patent-related activities. In Lannard Co. Inc. as a manager in product development and technical services, he was responsible for successful and compliant technology transfer of over 20 commercial products. Currently, he holds the position of Senior Associate Director, Product Development, Global R&D, Bayer Consumer Healthcare, USA. Here, he holds the responsibility of managing Bayer's global innovation budget, supporting 20 pipeline projects, and leads the R&D team with his technical advice. To his credit, Mr. Debanjan Das has several patents filed in US on the formulation development of many drug delivery systems. He has published many of his research works in reputed index journals. He is also an editor and peer reviewer in many international journals. His focus is currently on introducing new-to-world dosage forms involving complex global cross-functional projects. We are very much delighted to have such a knowledgeable speaker who is well-versed in state-of-the-art technologies of pharmaceutical manufacturing. Thank you, sir.
Thank you, Dr. Karthik. Yeah. So today we have a very knowledgeable person. So I request Mr. Debanjan Das to take over the session.
Sure. So let me do one thing. I will drive the slides then.
Yeah, sure.
Okay. By the way, Dr. Karthik, thank you for the introduction. I forgot that what I did all these years. Thank you for making me.
Thank you, sir. It's my pleasure, sir.
Not a problem. Okay, so let me. I have to do the share screen, right?
Ah, yes, sir. Share screen.
Can you see now?
Yeah, yes, sir.
Okay, so I'll go to the presentation mode. Hopefully. Oh. But if I don't think you can. Okay, can you see now?
Yeah.
Yeah. Yes.
Okay. Okay. Thank you, everyone, for having me. And you know, as we have been seeing through my career, that I started with basic research, and what I felt was we do such a wonderful research in our time, in our labs, such great ideas, so many theses we write. But sometimes I feel like all this beautiful work that we do, you know, human society does not get the benefit of it because they're never commercialized. People, you know, doesn't reach to all the people. So then I kind of move my focus more towards commercialization. So how I can do the work in the lab and in such a way that someday it can reach the masses and benefit, all the people, poor people, rich people, whatever. So that way it is a very, personal satisfaction, so that all the work. So that's why I kind of move more towards manufacturing from basic research, kind of bridging the gap, because I worked in the bench, I worked in the commercial field. So then I felt like, how can I bring the lab work to the commercial area? And that's why my QbD, which is a very important aspect in now manufacturing situation. So what I'm going to do is, I'm not going to go to the introduction. I mean, Dr. Karthik, thank you so much again. So just my career over the period of time, and USA, there are a lot of things happening. It's very competitive. So, I have to work in different areas, basic research, have to do work study, because, tuition is very high there, work in the morning and do studies in the night. And then different type of, I worked in chemical industry like Dow Chemicals, brand companies, generic companies, different types of companies. Contract pharmaceuticals is a big thing in USA, where they do contract research. Now I'm working with Bayer, which is a global giant. But basic science is the same. So, if you just know the science, it is the same working everywhere. So today I will just give you a little bit of how the product development goes, and then I will focus more on the QbD. The reason being, just I'm mostly a formulations guy, in pharmaceutics. That's what my training has been. So my idea is to just give you guys some idea about who loves pharmaceutics, and to convert other people who do not love pharmaceutics into pharmaceutics. So, just formulation. I'm not saying formulation is the best thing in the world, but pharmacology is also my favorite subject. But it just happened that, I came into this area because I'm more interested in making into the dosage forms. So, okay, so let's go ahead. Just to give you the idea of pharmaceutical product development, there are basically 4 stages, as you know: pre-formulation, prototype development and scale up, biological aspects, and commercialization. Pre-formulation, what you do is, there are 4 principal areas: physicochemical, let's see if I can move this. Okay. Physical chemical properties, bulk characterization, solubility, stability. I will not go into much details, but I think just that. You have to always start with your API, and you have to characterize very well. Otherwise, you cannot go forward. And industry challenges, solubility, of course, stability, definitely. So these are very important aspects before you actually even start to formulate. Okay, now different groups within formulation. So lot of different groups. So we start with biology. I mean, we start with so many compounds, and over the period of years, 10,000 compounds we start with. We begin with one compound, which actually makes to the market. Huge. Lot of work goes into it. Starts with biology team, starting out the pharmacology aspects, then chemistry team, how to kind of scale up bulk manufacturing. Then my thing comes how to make it into a dosage form, to take the molecule down in a dosage form. And then we work very closely with the scale up team, which we call we call it tech transfer, which basically just means transferring the technology in the bench scale to the commercial scale. So one of the important concept here is to understand when you are taking a molecule and developing into a formulation.
Excuse me, sir.
Yeah.
Can you minimize the screen?
Hello.
Can you minimize it? Control minus.
This one?
Correct only, madam.
It's done? It's good now?
Yeah.
Actually, it's okay, sir. It's okay, sir. You can carry
It's good? Okay.
Yes.
Oh, you were seeing that in the side. Okay, I'm sorry. Oh, all right. So yes, so coming back to that. So when you are starting with a formulation work, let's say even if it's a tablet or injection or parenteral, whatever it is, you have to see, I think it's very important that you have to see, especially in industry concept, that whatever you're making in the lab, it has to be commercialized. So you go to the commercial uh, site and see what sort of equipment is present there. Based on that, you do the formulation in your lab, because many times you will see you'll have equipment in the lab which is different from the equipment in the industry. So whatever you're making, it may not be translated to large scale very easily. So that is a really difficult thing, because then you cannot scale up, and if you cannot scale up, you cannot commercialize. And if you cannot commercialize, they will think that your project is maybe at your part it is a success, but in large scale it won't be a success. So it can so that's, I mean, from an industry perspective and a formulation perspective, that's a very important concept. That's why I wanted to share, because you start with something looking at the end in mind. You understand what is there in large scale equipment, then you start your formulation work in your lab. I don't know if I made sense, but that's what my experience had been saying. Like, end justifies means. Okay. So anyway, this is just a product development. You do API chemistry, pre-formulation, you type different dosage forms. And one of the important things over here is intellectual property. Remember, US is a free market. So any new drugs, they will do a patent, 20 years, but they will actually get about usefully maybe 12 or 13 years of patent protection. And with the patent protection, they will try to make up for the amount of money that they put in. For example, they had the blockbuster drug, anti-cholesterol drug, anti-lipidemic, simvastatin, a very famous drug. So they, and all the drugs, and all the brand companies, there a lot of focus is now, you make the drug in such a way that they get the intellectual property protection, so they can make the money. So basically what it means for us is you have to manufacture very, very quickly, because the longer time you take to manufacture, you will lose the time on the patent, because once the patent expires, it will become generic drug and all companies can make it. So basically your scale up has to be done very, very quickly, and you cannot lose time. So that's another aspect that why manufacturing is, successful manufacturing is important. Okay. And biological aspects, I mean, you do all sorts of screenings in vitro. Nowadays, a lot of focus on in silico, different phases, which I'm sure you know about it. Okay, now the commercial aspects. As I said, I mean, my industrial training and other pharmaceutics training, a lot of things they have been putting it, whenever a new project comes to me, they will say, when can you commercialize? Even before the formulation has started, they will put me the pressure. I will give you 1 year or 1.5 years. I want the product in the market. Somehow we make it happen. So that we work in that much of pressure. And so we have to find out what sort of equipment is available, who will give us a supply chain, whether I will get the bulk material or not, what will be the price. So for example, right now, many actives and excipients used to come from China. Right now, there's so much of problem. So when the project comes to me, I say, okay, fine. If the active comes from China, can I have a secondary supplier in, let's say, Mysore, right? So, or in Andhra Pradesh. So can I have a secondary supplier? Because if there's a problem in China, can I get it from India? Or if, let's say, excipient cellulose is coming from Europe. If there's a problem in Europe, can I get it from another company? Right now, everything is coming from the different parts of the world. You have to put it together and then manufacture either in India or in China or in US, and then bring it to the US. So you have to keep all these complex things in your mind. And sometimes you don't even see that equipment, what is there. Let's say if you want to manufacture in Hyderabad, you don't even know the company. Sometimes it is possible to, the manufacturing will be in US, but raw materials will come from another part, and you don't know where it is coming. Sometimes raw materials change and you have no control, impurities come in. So you have to keep many different things in your mind, even before you start the formulation work, so that you can successfully develop, you have the plan to do the scale up in time. Okay, Now I'm focusing on the QbD because it is becoming so much of complex operation nowadays for the manufacturing that it is very, very hard. I mean, you have to remember for the cost cutting operation, as I just said, there we are sourcing actives, excipients from different parts of the world. Sometimes their grades change. Sometimes everybody's working worried about the cost. So they are trying to source cheap materials from everywhere, and sometimes grades vary. Sometimes equipment is not good. If it's manufactured in US, at least or locally, I can go and check it. Sometimes it is done overseas where I have no control. But then all this, and this is not just one company. Every company is trying to do cost cutting, brand generic alike, and then they have quality issues. And FDA realized this, and in about 2005, they embraced this concept, quality by design. They said that quality should be designed into a product. You cannot make a product in such a way that, and you test in the end and say quality, it's good or bad. You have to make the process in such a way that quality is kind of built into it. So they got together with the International Council of Harmonization, which we call ICH, and they have different guidances internationally. Located in Europe, so they just to see that its global operations can be synced throughout the world. The different guidance document, ICHQ is the most important one for pharmaceutical development. They have also risk management, quality system, and other drug substances for Q11. So you have the link, and definitely click on the link and read them up just for the guidance. And also there's a FDA's QbD background on how they started it. I put the link over there, so you can go there and read how it all started. Okay, so based on the quality, keeping in mind the quality objectives of the drug, because of this variability, and you have to remember this, where is it coming from? Patients actually depend on the medicine that they're taking. If their medicine is of bad quality, it's really bad, because somebody might die, or even worse, if you're, it might not have even the drug inside. There might be some impurities with. So quality is very, very, very important. It's a matter of trust to our patients. So that's why this whole aspect of it is coming from that safety and efficacy for our patients. Okay. So what it basically means, quality by design, is any process. You make it in such a way that your end product will always be very good. I mean, I give an example of cooking. Let's say cooking sambar. Just a weird example, but I will just give it. You have different raw materials, like you have curry leaves, you have tomatoes, you have onions, you have tamarind. So those are your raw materials, right? And just imagine them as excipients. Then you have the process. You cut them, you cook them, you boil them with water, add certain things for a certain amount of time, and then you get your end product, which is sambar. And let's say that's a pharmaceutical product. Bad example, but just to put it in a funny way. So what it does is, anybody who is making it, its knowledge is good, but you have to also need the experience to do it. And experience, what I mean by, you see the graph in your right-hand side, your whole thing is your knowledge space, the outside gray box. And then the light blue is, you know a little bit more. We come to that DoE later on. Then the the darker blue one is the design space, where you think that I can narrow down to this. And then the innermost one is called control space. So basically, your box of knowledge gets smaller and smaller and smaller and smaller, which means that you have the wide experience that you know that if I vary certain things, what could be the result? For example, you forget to put one main ingredient, right? Then the effect will be really bad. Or you don't, let's say, you don't boil the water to make sambar for, instead of half an hour, you boil for minutes. It's not going to be bad. So you focus on all your knowledge and kind of narrow in. And I know these terms might be a little bit different right now, but you focus on most important parameters, what we call as the control space, where you can control these variables to an extent, and you operate within that space, the innermost circle, to know that if you work within these parameters, you'll always get a good product. Your summer will always be great. So that's how the idea is. So you basically process your knowledge in such a way that your input, end output is always passing, always great. So goals of pharmaceutical product is product quality, process scalability to improve process scalability, to improve product development and manufacturing efficiencies, and to do a root cause analysis in case your product is getting bad, why did it go bad? So basically what it means is, earlier the concept was, let's say again, going back to sambar, let's say you're cooking sambar, you cook it and in the end, maybe it's not, tastes good. So what happened? Every day you cook nice sambar, but today it is not tasting good. What happened? So you taste in the end and find that it's not good. Rather than, and then you have to throw it away probably. Rather than do it in such a way that you control how you make it. You get your best onions, you get your best tamarind, you boil it exactly for the same time. So you focus on more on the initial steps on upstream so that your end product is good. Okay, so more from downstream testing towards movement towards upstream process control. Okay, so now I will introduce to some of the terms over here, what QbD design concept means. So what they call is QTTP, which means is quality target product profile. So if you have a product, again, I'll go back to that same example. So what is your target product profile? How will the product look like? Is it I mean, pharmaceutically, is it, if it's a tablet, if it's a suspension, if it's a liquid, if it's a parenteral, what exactly do you want to make and how should it behave? For a tablet, for example, it should disintegrate in a certain amount of time. Otherwise, there will be no effect. So this sort of things. And then critical quality attributes. What exactly, how should it perform? Like I was just mentioned, disintegration time, something like that. Then critical material attributes, for example, this focuses more on actives and raw materials and how they should be sourced and what quality they should have. And then the second one would be critical process parameters, is basically how you process it. If it's a tablet, how long you have to pay, what is the compression force, mixing time, blending time. So this sort of things. So I'm coming to that in details. So this is what it is. So you imagine a pharmaceutical unit operation, okay? And then you have the input materials, for example, your active, your excipients. Let's say if it's a tablet, you have cellulose, you have mannitol, you have disintegrants, you have so many other things. So you control the quality of your raw material. Just like, again, going back to your sambar example, if the raw materials, let's say your dal and your tomato, onion, curry leaves, if they are bad quality, then your product always will be bad. So you have to very, very carefully control the quality of your raw materials. So input material is very important. Then critical process parameters, how you're making it. You're chopping the onion in a certain way, you're chopping the tomato in a certain way, curry leaves, you're good quality. I mean, maybe you're chopping it or not. So you're adding other spice. So this is you're boiling it. So you control your processing time, how you're making it. And then output is critical quality attributes, that you get a product, the sambar, in such a way that they always taste the same. For example, the sambar, you might have a certain acidity level or taste. So you control that part very well. So it is you will find the critical quality attribute. Let's say if we go back to now to a drug product, that is say let's say a parenteral drug, quality attributes, it has to be clear. There should be no suspended particles. The pH should be in a certain level. The osmolarity should be in a certain level. So these are all function of this important unit operations of critical process parameters and critical material attributes. If you see, the concept is very simple. Depending on raw material, how good you get it, and depending on the process, how control you process, you'll always get a good product. So that is it's a very simple concept. Materials, process will always give you, if you control them, will give you a good product. Okay. Now, this is actually just for you to understand. I know I've been giving the kind of examples, but this is actually from the FDA website, and you will get two examples, one for the IR product, immediate release product tablet, and for extended release product, you will see in the left-hand side. So these two you can download, go to this site and download. These are very important documents by FDA. And why they gave these 2 years is because many companies were not understanding the concept of this. But even if they understand the concept, they don't know how to apply it to the actual production process. So this, if you go to the website, download these two documents, it will give you the whole pharmaceutical development report, what they call as PDR, which is basically, they will tell the whole formulation development report, essentially how to take that active raw material, how to process it, how to risk assessment it, how to go step by step, pre-formulation, formulation, manufacturing, processing steps, and how to control them, everything they will give. And just to, I'm taking just a little screenshot, but these two, and you will get the whole report from this website. So to consult it, it will give you a very nice idea about the whole process. What operations you need to do right from bench to at least to the scale up stage, how exactly product is developed. At least for the pharmaceutics part, you will get a very nice idea. Okay, now one part important is QTTP, where you can see dosage form. What is that? A tablet. So fine. What is the dosage design? How you should do is immediate release. As soon as I pop it in, it will dissolve. How you will take it? You will take it orally. What is your strength? That's a 20 milligram. Kinetics, here is your 1 QTTP. Immediate release enabling Tmax in 2.5 hours or less, bioequivalence to RLD, which is reference listed drug. So basically, it's and stability, quality product attributes. So it's basically container closure. It's telling you what are the requirements of the product. What is your target? It should have all these factors. So basically, you are defining the objective upfront. So this is like your compass. This is how I want the product to look when it is made. So you have the whole summary even before you start the thing. So it is just like a starting point. Okay. Then critical quality attributes. You had the, the target was given, but then how it should behave, the tablet, how the tablet or how the product should behave. For example, what qualities it should have. So let's say physical qualities. It should appearance. What should be the appearance? Will it have any odor? No, we don't want any odor. What about the size? What about the friability of the tablet? How do you identify it? Assay. We need always 100% without any question. We need content uniformity. You always need good content uniformity because, obviously tablets are making the drug should be there in the correct dose, correct level. Dissolution, we want them to dissolve. If it is like a rock, it doesn't dissolve, then it's bad because, the medicine won't be in your system. So degradation products, it should not have any degradation product because that's bad for the body. Residual solvents, water content, microbial limits. So now you go to the details of it and find out the very critical quality attributes of how the product should actually look like in terms of these factors. Most important thing, if you ask me just to pick one, I would say assay, and the second one will be degradation. Now these two are very, very, very important one happening in the industry. Assay has to pass, especially for potent drugs, where the drug API is a very small quantity, and sometimes we have seen that the mixing is not proper or something is happening if the assay fails. And if the assay fails, your whole batch, maybe $1 million of batch, will be just thrown away. We cannot use it. And the second most important thing is coming now today is degradation products. What is happening is the way they are making the APIs, obviously they do some organic synthesis or even the excipients, and these organic solvents, they are coming into the product and they're very toxic. So even at parts per, I would say parts per million levels, the FDA is not allowing it. So when we test it, we have to also study this. And to make the matters worse, we expose them for stability in 40-degree chambers. Usually at room temperature, you will not find it. But as soon as the temperature is elevated, all these degradation products is different degradation of organic solvents, N oxides and all these things come up. And if they come up, they're toxic. And again, you have to reject the whole batch. So this will again give you more details, what quality you should monitor in your dosage form, that your product should not fail. Because what it happens, again, if you see the monetary aspects of it, FDA doesn't want your product, or any company, let's say, doesn't want their batch to fail. At least $1 million or a couple of $1 million will be completely wasted. And there's no exception. If something fails, we completely trash the whole batch, because it's a matter of patient safety, right? So you control these critical quality attributes from like this, from this focus. Okay, now how you do this? There's some design tools. So as I said, prior knowledge versus public knowledge. You gain your own knowledge with your experience that what can go wrong. This will come from your experience. The more you work, more experience you will get. Then you do some risk assessment. There are a lot of tools which I will go into more details, statistical tools, mechanistic modeling. And now there's a lot of focus on something called as PAT, which is called analytical technology, which is nothing but as you process, when you do the processing steps, you basically put some controls into it so that your process is highly controlled. So again, I'll come into that in a little bit later. So this is the whole approach. If you ask the whole thing, this is the whole flow chart in one shot. So if you can see QTTP is again in the beginning, then you find out what are the critical quality attributes which I was just talking about. Product attributes, what the product will look like. Then you design the development the product, then the relationship between CPP. Remember, we had the flowchart before, unit dosage form, CQ and CPP, design space, control strategy. And here, as you can see, you are using the statistical tools, prior knowledge, risk management, DoE, multivariate analysis. So what they are focusing on, as you can see on the right-hand side, you do the process control in such a way that control every step, that as you're going from process A to B to C to E to the end product, let's say D, A, B, C's are controlled, then D will always be good. So that's the whole thing. You control your process like you're making a somebody. You control the process of making a somebody. You cut the onions in a certain way. You use the tamarind in a certain way. You use your dal in a certain way. You maybe pre-soak them for a certain amount of. So you control every process very nicely, so the end product will always be good. That's the whole philosophy of it. Okay. Now I want to focus on DoE because what it is basically. It's a statistical approach to say that the different factors, and here they've given the example of making a cake, if you see on the left-hand side. So what are the different factors? So if you want to make a nice cake, let's say, you need an oven, okay? Of course, obviously, something like your source of heat, oven. You need sugar, you need flour, and let's say you need eggs. So what are the different levels? So if you have an oven, what temperature are you setting it, right? That is important. What sugar, how much sugar you're taking it, how much flour you're taking it, how many eggs you're taking it, that becomes your levels. And then what is the response? So what you get as an end product is a beautiful cake, and it should have a nice taste, nice color, nice consistency. Obviously, if you have a cake which is falling apart, it's no good, right? So this is just to give you that example Very simple concept, different factors, different levels. And you can get the reference in the bottom, just to give you some idea about what the DOE looks like. So what DOE essentially means is you study the factors, you study the levels, and you find a relationship between all these things, almost like an equation. It's not say almost, it will give you an equation. It will tell you exactly which factors to vary up to what levels and to what response we give, almost to a mathematical precision. Okay, so it's very, very, very amazing thing. Another example that they've given here is you have, let's say, starting material, number one, you have the catalyst and you have temperature. And here you will see that how you should vary the factors, which is basically different levels, high, low, medium, that sort of thing. If it's high and low, it's two levels. If it's high, low, medium, 3 levels, something like that. So how you can vary these things to get always a nice product. So basically, as simple as like you have been making cakes, maybe many of you, based on experience. But if you see, if you vary certain things, let's say your temperature is not good, maybe you know making a cake and the power goes away, right? Maybe for 2 hours the temperature goes away. So your cake obviously not good. You know that by your experience. But how to put that mathematically is important. Okay, so you will know exactly what temperature you will need for what amount of time to get a good product. So that's what it is, basically. It's a statistical approach, and how simultaneous variation of these factors can yield to the result. It will give you the relationship. Now, it's a lot of mathematics involved, and it's if you are a statistician, they will understand this very, very well. And we are not statisticians unless one of you are a statistician. So what we do, we just go to them and ask for their help. So what they will tell you is that it's statistics and maths. They will try to put everything into an equation. And what we do go to them and ask for the help, and they say, okay, this is how I can give you equation. Because if it's a pharmaceutical product, and if you need a precise equation of how these factors are dependent on each other, and you want that to an exact science, how we can do that, how we can achieve that, is by doing some previous experiments and then using those values to come to equation, so that every time you do that, you'll always get the same product in the end. Okay, so it's basically prediction equations. If you, let's say, based on some pre-process input variables, which we call KPIVs, like input variables you saw before, and then it's a function of all these factors. So very quickly, I will just go through different aspects of you. This is a very big subject. Please take some time and study this. It's a very, very, very important concept. Everywhere industry is using it. We use it even in the bench in formulation development. We use it in scale up. So what it does is it's a very powerful tool to tell you that what not to do, or rather what to do if you always want to get a great product. And how you do that is making different extreme examples or extreme experimentations to give you the input that these are the inputs you're allowed to do or not allowed to do, so that you always get the good product. So as I was talking about, if I go back to it, say, remember this, these are called in that oven thing, these are called factors. What factors? And these are called levels, and these are responses. So again, levels and factors, always remember factor is to the power. Okay, level is, just to remember it, how you find out how many experiments you have to do. Let's say with 2 levels and 2 factors. So level is in that, factor is in the power and the top and the level. So if you have 3 levels and 4 factors, is 3 to the power 4. So if let's say there are 3 levels, high, medium, low, and 4 factors, exactly as you had before, like oven, sugar, flour, eggs, you will have 81 experiments that you have to do, 3 into 3 into 3 into 3. So you have to do so many experiments to get to that equation. So as you can see, there's a lot of equations, right? A lot of experiments to do. Again, there's another way people do, it's called one factor at a time. They, again, many experiments. So the whole idea of, and then full factorial, I'm sure many of you know, it's the same thing as I just said. If you have 9 factors and 3 levels, 9 into 9 into 9, the example that you have in your right side, let's say a simple thing like a tire, you can go that in details later on. So basically and then there are, there are some complicated designs. So what the bottom line is that, you know what the factors important, you know what levels you try, but do you really have to do so many experiments? I mean, really, if there seems to be a lot of experiments to do, it will take a lot of time, a lot of resources. So these DoE tools, this is the most important thing, and this design will allow you to do very less number of experiments and will still give you the correct equation, or equation with almost 99% correctness. That is the power of the DoE. That is how you increase efficiency. Instead of blindly doing so many experiments to find out what can go wrong, what can go right, or to get to the equation, you cut down your time to reach at your desired location. You treat them statistically so that you get a result by doing less number of experiments. Okay, so you know exactly how many, if you're making that cake again, you know that instead of doing like so many experiments, 9 into 3 factors, 4 levels, that is 4 to the power 4 to the power 3 to the power 4, so many different experiments, you cut down to a very less number of experiments and you still get to the same results. There are some terms you will find that Taguchi's design, Plackett-Berman, response survey methodology, which is now widely very used. So these are different types of DOEs. So read them up in your spare time. And what we very much use, people use in the industry is these one. One is, sorry, Taguchi's design, Plackett-Berman, response surface methodology, and underside there's a box Benken design. So again, it's really statistical treatment of different things. All you need to know here is that the whole idea is to not to do experiments as here. So if you have, as I said, if you do a full factorial, so the whole idea is you cut down your number of experiments needed to be done to a very minimum. So from 4 to the power 4 or 3 to the power 4 experiments, 81 combinations here, maybe you have to do like just 15 experiments and you will still get the result. So basically use less time and you get the result. So please read on the DOE part. It's a very important concept. The whole idea, remember, is to give you the equation by doing less experiments so that you get the complete idea of how your process or product is going to behave to give you the right product in the end. Okay. So that goes on the initial processing part. Now I will come to another concept, which it's a lot of traction in nowadays. It's called process analytical technology, which is nothing but you control your process all the time. Let's say if it's a simple process of mixing and boiling, right? How long do you mix? How do you mix? And how what is the product behaving in that time? If you're mixing for 2 hours, what we do is, there is a very important concept. So I'm going to do this. If you can read in the bottom part, at line, online and inline measurements. What it means is, let's say you are trying to dissolve something by heating. We'll call it boiling, for example. Now, is it really dissolving or is it something else is happening? So in that, while the process is happening, while it is, let's say, boiling, you try to measure what is happening to the drug inside. So something called as at-line measurement. So basically what you do is you measure, you take the sample and you have the measuring equipment, let's say spectrophotometer. You immediately take the sample, analyze, and tell, okay, has it mixed properly? Yes or no? So you will get the result right away. If you have, so what it does is you have to know, okay, now we still have to mix it for another half an hour or boil it for a half an hour, something like that. So we'll keep on, and you find out the endpoint like that. Then the next part is called online measurement. So what it does is, in the process, what they do is they will take a sample from there. It will divert the sample automatically, and there is inline, there will be some either spectrophotometer or NI or whatever it is. It will tell online, online, it will tell it will give you the result, and it will put the sample again back. So it is almost like a real-time, semi-real-time process, which is telling you what is happening to a formulation when you're exposing that to the process. And the latest one is you have, and this is my favorite, they will put sensors inside. Either it is a powder blending operation, or if it's a liquid boiling mixing operation, they will put sensor inside, and they will tell you exactly, what is the situation happening inside your reaction vessels. If it's a blender, or here you can see that's a fluid bed over here, they'll put sensors inside like a Raman, and one second. They'll put a Raman over here, and NIR. These are the two ones which I have used. So what they will do is, when you are doing the process, when you are mixing it, when you are fluidizing it, what is situation? Is the most boiling and the liquid is still simpler than powder mixing, which I find most challenging, especially in potent drugs. Little bit of the active and a lot of other diluents, and a lot of problems with the mixing. They never mix. Sometimes they disaggregate. So drug is on one side, excipients on the other side. And when you compress them, you will have tablets where there is no drug. Very, very big problem in the industry. So we put a lot of focus during blending and fluidizing bed operations, where you need to know that the mixture or the blend that you're using is uniform. So you put in the sensors right there, and then you find out in the real time, while the mixing is going on, whether it's an uniform blend or not. And how you do that? You take an NIR spectra. And NIR spectra, you first know that if the uniform blend, how it looks like from your library, you first do your control graph. And when you go to this, let's say this is a 1,000 kilo batch, and you keep mixing till you get the graph which shows that it's uniformly mixed. So then you know that, okay, now you know that's the DOE. If I mix for, if I rotate it for this or fluidize it for this much, for this amount of time, I will get a very nice uniform mixture. Okay. So basically, if you see here, it's very important when you do PAT in process design, scale up, and commercial. So if you do this, if all these things are what it's doing is, it is controlling the process. Mixture is uniform means what? When you compress the tablet will always be uniform. So your end product will always pass. This is process scalability. I will not go into much details, but this is again, this is just different mathematical treatments. It will tell you some mathematical ways how to say that all these process, scale-up processes are always under control. Different, it's a matter of there is the product and process understanding that you gain in pharmaceutical development. So the difference between this and non-QbD approaches, in non-QbD approaches, you always do the testing in the end. You taste the tablet or you taste the liquid, whatever, and you know that whether it is passed or failed. Here, you're continuously testing it while the process is happening. So that's and it's telling you how good your process or how good your process capability is, and which will lead to a continuous improvement. For example, let's say your blending process, you do these calculations and find out I'm blending it for, let's say, 2 hours. However, when I'm doing this online at-line measurements, spectra, maybe if I mix it for 1.5 hours, it is good. Provided if I mix it very fast, maybe 1 hour is also good. So all this information will give you how to have a good control on the process and how I can continuously improve it. And the concept is important because in industry, you are paying by the hour. Big equipment, a lot of people involved with mixing. So always they want to cut down on the time, how quickly you can make it to get the good result. So that's the basic underlying philosophy behind it. Okay, now the control strategy is basically, it's very simple concept. Level 3, as I was saying, level 3, level 2, and level 1. Level 3 is the end product testing, which the whole company used to do end product testing. Now level 2 is in product and some in-process testing, and level 1 is, as I said, you just do while the process is going on. It's a process control. So right now, what they do in most industry, they are going towards a hybrid of level 1 and level 2. You control some of the process and also you control the end product. So the ideal one would be level 1, as I was just telling. You continuously control the process, but you have to remember one important thing here, that going to the level 1 is actually expensive, because remember, level 1 is we are controlling the process in while it is happening, right? We're inserting probes into it. We have to hook it up to an NIR spectra. We have to compare those things. So it takes some investment. It's not something like testing an end product where you take the tablet, throwing into HPLC and get the assay results. So it's not as cheap or easy direct as that. You have to involve some brain power and some money into doing this in-process testing. So many companies are going towards it, but they are not there fully. But the ideal would be to go to level 1, where it will tell you, as I said, the whole process control as you're making it. Okay, so but one-way investment is good, because if you invest it now, product will never fail. So one way, it's just like how much money you want to put forward, to see the end result will always pass. So this will give you the summary of the whole thing. So you remember we started with quality product profile, what we need the product to look like. Then the process outline, prior knowledge, process development, processing design space, control strategy, and continual improvement. So as you can see, the green line on the bottom is development work, and then manufacturing starts. But one important thing over here is, in the development, if you are not having these things in your mind, right, then you cannot go to the manufacturing step, because you will see at one point, the green line and the blue line in the bottom, they're overlapping. So if you have, the whole idea is, you start with your end in the mind. You develop a process which you will know that will always pass. You don't make a process in such a way, it's so different that when it goes to scale up, you will see process development, process design space, that it's a different thing, it will never be scaled up. Let's say practical example is, again, I'll go back to tablet. You will use a punching machine in such a way, or blending in such a way that it is good in lab scale, okay? It's maybe 2 kilos of the blend, always passing, nice mix. When it go to 1,000 kilos, what will happen? My goodness. Oh, it's not mixing properly, or it is desegregating. The tablet that you are making is capping. Its friability is really bad. So you have to know in such a way. And then again, the whole concept of your material comes in, your process comes in. So how you're getting the material, is it good? How you're getting the process, is it good? When the process that you're using in the lab scale, can it be translated into large scale? So you have to keep that in mind. And all these things you can do, all this control, by doing these experiments, doing the DOEs, doing the process analytical technology. You can use these tools to always know that your product will follow a process, and the product will following the process will always pass. Okay, now coming to the conclusion. always make the formulation with the end in mind, because eventually, as I said, you have to, make the product, you have to sell the product so that it will reach everybody, not even from the profit perspective. Because I'm an R&D guy, I'm not a CEO of a company, but just for me, the satisfaction is the product that I made here, even a simple tablet, is reaching everybody. And how it can reach everybody? Only if it can be commercialized, right? Otherwise, how will it reach everybody? You have to know your equipment and machines very, very well. Not your lab skill, but your industrial skill. There has to be a balance, same thing between the two. You look at the bigger machines, then you try to get this machine smaller in size in your lab. Sometimes it is not possible, I totally understand that. But your attempt should be like that. Or try to find out the basic principles of scale up, bigger machines, and use the same principles at least in your smaller machine. Otherwise, you make it in such a way it can never scale up. Okay? Make sure that your ideas turn into a product. This is the most important thing. Otherwise, as a scientist, you'll not feel, you will not feel accomplished. That all the effort that you're doing in the lab, if it is not being scaled up, you will not get the satisfaction. And how will you get it? If it can be only scaled up and commercialized. Okay, so that's my whole idea. End justifies the means. You know what is happening in your end stream, and then focus based on that in your upstream process. So that's the whole philosophy. You develop everything keeping the end process in mind. You start now, not looking at the end. You first study the end, then you begin, take the knowledge and start in the beginning. Okay, and that's all I have for you today. And now I'll open the floor for questions.
Yeah. Thank you very much.